So let's get started, get this on the way. So it's my great pleasure to welcome Elizabeth Reed to talk in our Theory of Living Systems seminar today. Elizabeth has a background in physical chemistry and I think chemical engineering. And over the past sort of a good 10 years or so, she started to combine mathematical modeling with an engineering perspective, applying this to biochemical and biological systems, really from the scale of molecules all the way up to cells. And in her talk, she will do something which I think is amazingly promising, to combine multi-omics with mathematical modeling. And I'm very much looking forward to hear Elizabeth talk. I've heard her before, and I know how much I've learned last time. So I'm looking forward to learning even more today. And Elizabeth, the stage is yours. If people have questions, please put them into the Q&A or chat. I will try to monitor them. And after the seminar, we will either read out or we will unmute people to give these questions to Elizabeth. But now, Elizabeth, please take to the floor.
Great, thank you so much, Michael. Thank you. So I'm very excited to be able to give this talk today. So thank you very much. And as Michael mentioned, I'm going to be talking about trying to combine stochastic mechanistic models with omic types of data. And we have sort of two different areas where we've been doing this. And so first of all, I just want to start with what is my motivation for focusing on stochastic models. This is an image from a review article that focuses on stochastic gene expression, which is revealed here beautifully by these different colors in a variety of different cell types. But for me, my interest in stochastic fluctuations comes from the fact that, first of all, these become so significant when we're talking about low copy number events. And that's why famously stochastic fluctuations are thought to be so important in gene expression. And there are also instances where these are thought to have real significant sort of downstream consequences for cellular behavior. And I've listed out a couple of those here. But maybe even more relevant for today's talk is that we increasingly have available high resolution data. And by high resolution data, I mean a number of different things, maybe single cell data or single nucleotide data, single molecule types of data. And all of these types of data, of course, reveal heterogeneity, reveal distributions. And so my group is interested in building models that sort of have those in them from the get-go so that we can compare those types of models to the data and see what we can learn from that kind of approach. And so I'll be talking, I suppose, broadly speaking about epigenomics, and on the left is the famous Waddington's epigenetic landscape, representing cell state as somehow existing on this potential surface. And here I have two broad different definitions of what people sometimes mean by epigenetics. So the top one up here is maybe an older definition, which is epigenetics in terms of transcriptional networks and dynamic systems. So the idea that these different cellular states, these different phenotypes that cells exist in, are really resultant directly from different regulatory interactions in these complex dynamical systems of gene regulatory networks. Another definition, and probably nowadays at least more common definition of epigenetics, is that of chemical modifications that are occurring either to DNA or histones associated proteins. And of course, these work together with transcription factors to regulate gene expression as well, and to regulate the structure and compaction of chromatin. And so today I'll be focusing mostly on DNA methylation as an example of one primary epigenetic modification. So these are two areas that we've been working in, and I am going to describe a project in each of these, starting with this area of epigenetics in terms of DNA methylation, and then going into the gene networks definition of epigenetics. Okay, and so in this project, we've been interested in learning about the kinetics of epigenome replication. So what I mean by that is that every time DNA is replicated, we often maybe forget that not only does the genetic code have to be replicated, but actually the whole epigenome essentially needs to be rebuilt as well after the DNA is copied. And so As I said, we're focusing primarily on DNA methylation, which is one ubiquitous and well-studied and fairly well understood epigenetic mechanism. And so the primary sort of location in our genome for DNA methylation is on CG, or cytosine guanine dinucleotides. So you see these dyads provide this symmetry. So the methylation is occurring here on the cytosine, if you can follow my cursor. And so what happens is that you have, generally speaking, symmetric methylation across DNA. When DNA replicates, which is represented here in this cartoon, initially, after the nucleotides are added, they're unmethylated. The cytosines are unmethylated. And so then what you need is for the DNA methyltransferases to come along and add back in those methylation, those methyl groups. So what we're interested in here is how rapidly does that occur? And can we learn something about the dynamics of this process? And so we're working with some experimental data that comes from some recently developed techniques. So this is work that came out of the Meissner lab. And my colleague, Tim Downing, who's now an assistant professor in UCI Biomedical Engineering Department, is pioneering these methods in his lab now. And so the idea was that they found that if they could measure methylation on these cytosines, or these cytosine guanine dyads, if they could measure this across the genome, which one can do using whole genome bisulfite sequencing approaches. But then if you can do that in a way that's associated with DNA replication, then maybe you can look at how, what are the dynamics of replicating that epigenomic information. So the idea is that they use a nucleotide analog in order to track time post-replication and look at the methylation state. And you see things like this. So this is just averaged over the entire genome. So we're looking at something on the order of 20 million cytosine sites and averaging the level of methylation in post-chase experiments. So this is hours up here. And this is the mean CpG methylation on the y-axis where my cursor is. And so following that through with time, it starts low and sort of goes back up to what we call bulk levels or average levels. Another way of looking at that is the plot up here, where you have the green curve is showing methylation across a certain portion of chromosome 17 in the genome. So that's the green. That's what you would typically get out of whole genome bisulfite sequencing experiments. And you can clearly see that there's these dips. So there's a very bimodal, there's known to be a bimodal distribution where you have these very demethylated regions and other regions where there tends to be relatively high. This is probability of methylation of a site within that region. But the other thing to notice here is that in nascent DNA, so shortly after replication, it's lower, showing that those levels of methylation aren't quite back up to bulk levels. And as I mentioned, this is with Tim Downing's lab. So we were interested in applying statistical inference approaches to these data in order to see what we could learn about the kinetics. And so we started with a very simple model, which is just our initial assumption that each individual site is an independent stochastic process. So we can fit these time points that we have, this time point data. Each site is described by a rate, K, so that's what we call the rate of remethylation at a certain site in the genome, and a fraction F, which is representing the steady state fraction of cells that would be found methylated at that site. So the simplest possible model really that we could come up with, and turned out to still reveal quite a lot for us. And so we're using here maximum likelihood estimation to fit these parameters for, as I mentioned, these roughly 20 million CpG sites across the genome. And you get things like this. If we look at a representative site, it looks pretty noisy, but we can also see that this model is doing what it's supposed to do, which is trying to fit this, monotonic exponential model to the data points that we have. One of the things that characterizes this data is that we have very different sampling depths at different sites. Some sites we happen to get hundreds of samples, and that's great. Other sites we have really only a few, 10 or sometimes even fewer. And of course we have relatively few time points. We have here, this is showing 4 time points that we have. Sometimes we get 5 if we're lucky. So coarse time resolution, sometimes poor sampling depth. The only advantage that we have going for us is that we're looking at the whole genome, or specifically, as I mentioned, these, depending on your read depth that you take, between 10 and 20 million CpG sites. So that's the advantage that we have. So this work in my lab was started by Luis here, who was a master's student at the time. Now he's a PhD student at University of Chicago. And so one of the things that we can do with this kind of fit, if we, once we compile basically or estimate these parameters across the genome, is that we can start to separate out different kinds of kinetics. And so as you might imagine, if we have a representative site that has kinetics like the orange curve, I would call that a high F, but a low K site, so relatively slow to reach steady state. But once it gets to steady state, it's 100% methylated at that site. That's in contrast to the yellow curve, which would represent a low F but high K. Another way that we think about that is that these sites that maybe look more characteristic of the orange curve are sites that are going to exhibit more temporal heterogeneity, but less cell-to-cell heterogeneity in terms of the heterogeneity that we might see in these kinds of data. And this was significant, we think, because of the fact that the temporal aspect of this heterogeneity may not be as highly appreciated as the presence of cell-to-cell heterogeneity in methylation levels. And then it's important to point out here that in terms of more traditional, or what people call bulk whole genome bisulfite sequencing measurements, both of these sites would look identical, these representative sites, because they have the same time average. Okay, and so I also want to mention that we did worry and think about whether or not this 2-parameter model, which is just completely monotonic exponential, does a good job of describing the data that we have. Another thing that we can do is add a 3rd parameter and look at reversible kinetics or non-monotonic kinetics. And we certainly can find sites in the genome. These are randomly selecting 4 just to see what they look like. We certainly can find sites that look characterized by these kinds of reversible kinetics. We found overall that the majority seem well described. So far, we've looked at this just according to a BIC. The majority of them seem well described by the monotonic kinetics. The ones that aren't well described by that look really intriguing to us, and we have further work to look into those and see what's happening there. Are they, are they being proofread? So these are histograms that just show the distributions that we can get. These are from chromosome 1. This is the distribution of parameters. Again, so these are single nucleotide parameters, inferred parameters. For the rates here shown on a log scale, and the fraction of cells that have that level of methylation per site. And one thing that we thought was interesting was just the range of timescales that we find here. So just trying to pick apart this distribution and put it into more understandable numbers here. We have a median that's somewhere around 1/3 of an hour for the median rate at which these sites recover steady state. But we also have certainly sites that seem to be remethylated in a matter of maybe several minutes. We also have some that are very intriguing because they have much longer timescales of remethylation. And these ones down here, for example, if it has a 4-hour half time to reach steady state, that is spending a significant fraction of the cell cycle in sort of not its complete sort of steady state level of methylation. The other thing that I want to point out here is that the distribution is cut off here, and that's because we're reaching the limit of our experimental time resolution to be able to infer rates any faster. So in reality, many of the parameter fits here on the edge. We're trying to estimate a lower bound on what that kinetic rate constant is, but we can't identify it. So the next thing that we looked at was, could we look at the correlations of these remethylation rates across the genome? And also, could we look at that in a region-specific way, looking at what the local environment in the genome, how does that affect the correlation rates? So initially, we looked at like this. So what is being plotted here is on the y-axis, this is the correlation, the neighbor correlation of rates. So if one site is has fast kinetics of remethylation, how likely is it that its neighbor also has fast kinetics of remethylation? And on the x-axis is just plotted genomic distance in base pairs. And these are looking at a tile from 0 to 1,000 base pair, or a distance of 0 to 1,000. So the genomic genome-wide correlation function is plotted here. But what we found was that when we start to filter by different genomic regions, we have quite a bit of variability in terms of the shape, the length, even individual features. And this is still something that we're actively working on analyzing. But we've done a little bit of work so far. And so, for example, if I just pull out a couple of examples, I'm looking here at CGIs. These are so-called CPG islands. So these are regions that have relatively higher density of CPGs. And so these ones have a correlation function that tends to be strong. It tends to last a while, and I know it's cut off here because we're only plotting to 1,000, but these tend to have relatively strong correlations compared to something like the line elements here, where this is representative of a region where the correlations fall off relatively rapidly and are then quite close to 0. And so if we just look at what other things this correlates, what does the correlation correlate with? One thing that we found was that regions that have this high correlation, we also think of that as having more collective methylation dynamics. So so they tend to sort of all go fast or all go slow in these CGIs, at least relatively more than other places. So we found that places like that have both relatively higher chromatin accessibility as measured by DNase level, but also relatively higher CPG density. So those things are all associated. This is work by PhD student Honglei Ren. And the next thing that we wanted to do is we wanted to see if we could learn more from these data and these statistical inferences by combining with some kind of mechanistic modeling. So the idea here is simply, here's our kind of data analysis or statistical inference pipeline from the experimental data. We want to be able to generate in silico data according to different types of hypothesized mechanisms, and then put that simulated data, put those through the same analysis pipeline with the same poor time resolution and issues with sampling depth and so forth, put those through the same pipeline, and then try to compare these outputs and see what we learn. And so here are the types of models that we've looked at so far. And so I'll try to go through these relatively quickly, but the idea here is that these are stochastic biochemical reaction network models. The one on the left is essentially a Michaelis-Menten type of kinetics, but where we're simulating this with, on the single molecule level. So we are tracking, individual DNA methyltransferase enzymes in the simulation, and we're modeling a substrate of DNA, and we're treating this as just a one-dimensional grid here, and tried where we could to extract the parameters for these reactions from the biochemistry literature. It's a little bit difficult because those are done in purified systems, and what we're looking at here is data that comes from the cell. So we're not sure how well we know all these rate constants, but we tried to do that where possible. So the other two mechanisms, this is a distributive model. So it's one where we have just complete independence. The enzyme binds at a certain site, carries out the reaction, and unbinds. We've also looked at a couple of additional models. One of them is called a precessive model or a diffusion-based model. So the idea here is that the enzyme binds to a hemimethylated CPG substrate, but then can also move by one-dimensional diffusion along the DNA. And then thereby reach nearby sites. Another model that we looked at has sometimes been called the collaborative model or a recruitment-based model. So the idea here is that we also have interdependence between the sites. But here, what happens is that when the enzyme binds at a certain CPG site, it just increases the propensity of binding of another enzyme to a downstream site with some sort of distance function that we essentially have to make up, some distance function by which that falls off, that increased propensity. So these are the types of simulations that we've done, and they're somewhat straightforward because for the most part we can do kinetic Monte Carlo, or otherwise known as SSA or Gillespie simulations for these. It's a little bit more difficult to deal with the diffusion because it gets really expensive if we have to wait for the enzymes in our simulations to do all their sort of back and forth, individual random walk steps. So we adapted a first passage time method where we can sort of speed this up because we only care about what's happening at the CpG substrates. So this is just a snapshot, and I'm not going to go through the details because I want to get to another project as well, but this is a snapshot of the kinds of outputs that we're looking at from our simulation. So let me just walk through what we're looking at. On the left-hand side, we're looking at the collaborative model. On the right-hand side, we're looking at the processive model. I'm realizing now that order is switched from the previous slide. I'm not showing here the distributive model. You might intuit, and you'd be right, that the distributive model doesn't show correlations. Okay, and so for that one, we just sort of can cut that one out right at the bat because it doesn't show any neighbor correlations, which we know exist in the data. And so what we're plotting here is all the black curves are these experimentally derived correlation functions. Here we've zeroed in on, I think, 50,000 CpG sites in each of these different regions. Up here are the CpG islands, then the enhancers and the lines as 3 representative genomic regions. And then we're looking at different simulations. We have not done an extensive parameter optimization. So we're looking here at just sort of different sort of sampled parameters. And the only real takeaway from this slide is that in general, we can sort of match the shape reasonably well, depending on parameters like, for example, the diffusion coefficient in our processive model with a few fit parameters. And so maybe this is one way that we can try to connect our models to the data. However, we have a lot of unknown parameters in these models. So we looked for something else, another way that we could potentially discriminate between these two models. And this is work in progress, but so far we think we have found one thing, and that's kind of, I'm not showing the results, but I'm summarizing them here on this slide. So the idea is that, as I mentioned again, this distributive model, it does not show any neighbor correlations. The neighbor rates are not correlated. And so we just forget about that one. For this processive or diffusion-based model, the neighbor rates are correlated, and we see these regional differences in correlation functions that we also see in the data. However, what we see is that the correlation does not strengthen with increased CpG density. It actually shows the reverse trend. And that's in contrast to the collaborative or recruitment-based model, which does show the sort of correct trend in terms of how the correlation depends on CpG density. And so what we think is happening, or we know what's happening in terms of the models, is that the recruitment model or collaborative model simply just does exactly what it does. It increases the propensity of recruitment to the region of additional enzymes and gives you longer correlation lengths. The processive model does something different. When you increase the density of CpGs, you squeeze more of them into a region, you're actually increasing the stopping frequency of one enzyme as it tries to move along. And every time it stops, this enzyme has a relatively slow catalytic rate, and we think that that's actually leading to memory loss in that catalytic step. And that leads to a shorter correlation length. And so to summarize these results, at this point, we think what these simulations are pointing to is the fact that the correlations that we're seeing in these genomic data are more likely resultant from this kind of recruitment-based model rather than a diffusion-based model. Okay, so I want to just sort of summarize. This is ongoing work that we're working on, but in this work so far, as I mentioned, what we can do with these statistical inference approaches is measure kinetics with nucleotide resolution. And what's exciting about that is that we can do it in cells. And so we can get kinetic data, even though we have poor time resolution, we've got the whole genome. So we have sort of a richness of data there that we can work with. And one of the things that we've learned so far from those sorts of inferences is that we have this interdependence between CpGs. And by comparing to the models, we think that in general, we still think that the processivity is occurring. In vitro studies have shown evidence of processivity of this enzyme, but we think it's a less significant factor in giving rise to the sort of regional correlations that we're seeing. The reason why we think this is potentially significant is that there's been mathematical modeling work looking at how the collective behavior or interdependence of CPGs is actually really critical for the stability of the overall genomic methylation patterns. If you just have independent sites acting by themselves, and you have just a little bit of an error rate in terms of whether they're going to be correctly remethylated every cycle of DNA replication, you're just going to lose your whole methylation landscape, or your methylome, as people call it. And so the direction that we want to go with this work going forward is, in addition to trying to look for ways to validate or invalidate our results that we have so far, we want to look at what the potential significance of these sorts of differences in mechanisms might have for sort of regional differences in stability of the epigenome, and something known as epigenetic drift, which is changes that actually occur in humans and over their lifetime. All right, so with that, I would like to move on to the second theme. So I am switching gears, and I want to, before I go on, the connection that I want to make here is that even though these are two disparate areas, and they're two different kinds of quote unquote omic data, the theme between both of them is we're trying to look at whether or not there's information in these omic data that can, combined with stochastic models, reveal new insights. And so that's the connecting theme. And so the second theme is looking at the question of how much mechanistic information can we get by looking at fluctuations in gene expression, especially in light of the sort of new high throughput omics, transcriptomic data that we have nowadays. And so what I'm showing on this slide is a couple of quite sort of classic examples of previous work in this area where theory of stochastic models of gene expression and gene regulation compared with distributions showed some insight. So this figure here is from a review, this review article by Arjun Rajan, Van Oudenaarde. This is also a really nice review article by Munsky et al. on this type of topic. So in this example, the idea was that the shape of single gene expression distributions reveal whether a gene can be, whether a gene has one or multiple activity states. One state is like an unregulated promoter. More than one state might reveal a regulated promoter. And in the second example over here, this is an example where the kind of feedback that's occurring of a transcription factor on itself, in this case, negative feedback, negative autoregulation, diminishes the fluctuations, decreases the fluctuations that are measured. So these are both examples of how shapes of single gene distributions can reveal information about what's going on in gene expression. What we really want to do is to try to extend these kinds of concepts to multi-gene distributions, because that's where we can really take advantage of high throughput data sets. So here I'm showing an example of single cell transcriptomic data. This is data from this paper in Science from 2018, and it's looking at a snapshot of frog development. And of course, we are often used to now seeing this incredibly high dimensional data being plotted like this in these projections, this SNE plot. So this is single cell data in a high dimensional gene expression space on the left-hand side. On the right-hand side, I'm showing single cell data in a low dimensional gene expression space. What do I mean by that? This is just looking at a pair of genes, just two genes from the tens of thousands that have been measured over here. And so this is looking at RNA counts. These two genes are called SOX2 and T, brachyury. And this is from developmental stage 10. But the point is that we have this interesting looking shape of this distribution where the yellow is showing a large number of cells that have that combination of RNA counts. Okay, so you see a lot of them tend to be either low in one or low in the other one. And then out here in this region, we would have cells appearing that have some amount of co-expression of both of these two genes. So we're going to be using these kinds of 2-gene distributions. I sometimes refer these as gene co-expression landscapes. And so this is going to be what I'll be showing a lot of from simulations for the rest of this talk. So the same kind of way of plotting these distributions. So again, our question is, can we kind of extend those ideas from earlier work about single gene distributions and inform our understanding of interactions from single-cell RNA-seq data? The challenge, as always, is the curse of dimensionality, especially when we think about trying to develop mathematical models of gene-gene interactions. What we have in principle from these data are hugely high dimensional. And so to even sort of the space of possible models, the space of unknown kinetic parameters just becomes overwhelming. Not to mention the computational challenges with trying to compute these models, which is a challenge for discrete stochastic models. And so in the rest of the talk, I'll show what our approach has been so far to try to address this. So I'm going to start with a class of models that we've been looking at. And this is a fairly standard kind of form of a 2-gene interaction model. So if I call 2 genes in a mathematical model X and Y, and the types of interactions that we're considering are the interactions between them going in both directions, and also the self interactions. And this little notation here just means that each one of these 4 interactions could be considered to be either activating or repressing or just not interacting at all. And so we're going to be interested in looking at these gene pairs, partly because we think they're an important building block for building up ideas, information about complex networks, but also because it's much easier to think about going from single gene distributions to 2 gene distributions and not all the way to n gene distributions. And so this is just representing, if we compute say the steady state probability distribution from one of these 2 gene models and project it into one dimension like this. This is a probability distribution. We often are going to be plotting what is technically known as the quasi-potential distribution, which is just this log transformed probability distribution, so that it looks sort of like a landscape. All right, so where we've got our wells being like potential energy wells, being sort of our modes or in this multi-stable system. And this is what it looks like as, for one of these potential models where we're plotting, again, this quasi-potential landscape. This is the steady state solution. So what we're doing here is we're numerically solving a master equation, a chemical master equation, for the model that I showed up here and didn't really describe yet. I'll do that on the next slide. Oops, sorry. The first thing I want to say is that, again, these go back to the multi-state models of gene expression, which are highly, highly simplified models of gene expression, but nevertheless have been done quite a good job, I think, of describing gene expression and gene regulation, going all the way from prokaryotes to eukaryotes. So the idea is that you have 2 or more small numbers of states of gene activity. So you've got an inactive state where it's not expressing, an active state where the gene is expressing. And in this case, this is tracking just sort of production, as in this model, transcription and translation, everything are sort of subsumed into a production rate and a degradation rate. So that's really the backbone of the model. Even these simple models can have quite rich dynamics, and there's been quite a lot of theory in this area. The dynamics are interesting because, for one reason, is that the kinetics are thought to be potentially very widely variable for these switching rates. So for example, if you think of the switching rate between inactive and active states of gene expression to be due to something as simple as a single transcription factor binding, then those could be very, very rapid. On the other hand, if you think about it modeling some much more complicated chromatin remodeling process, as diagrammed here, then that's can be orders of magnitude slower. And so in these highly simplified models, we try to run the gamut of sort of treating those various regulatory modes by changing those kinetics. Okay, and so when we link up 2 genes of that mode together, it could look something like this. So again, the cartoon here is really based on the last ideas, where we've got transcription from both of these genes. We've got degradation. Couple of additional details here is that each of these genes is often modeled as forming dimers, homodimers, and this is a way to take into account cooperativity in terms of regulation, which is often measured, but that could be considered to be a parameter of the model. Maybe you don't have cooperativity, maybe you have trimeric regulation or otherwise. And for this kind of 2-gene model, in principle, we consider the 4 different binding states. So if you're looking just at gene X, it can have binding states where itself and Y, neither gene is bound, where it's just self-bound, just other bound, or both bound. And then we assume that the transcription rates from those different states vary. And so depending on how you vary those transcription rates based on the binding state, that's how you encode a certain network topology. So in other words, up here, the various network topologies that you can have here are encoded by these transcription rates. And the one shown here is representative of a mutual inhibition and self-activation type of network. Okay, so with that, so we build these stochastic biochemical reaction network models. And we try to vary the parameters in plausible ranges. Things like transcription rates and degradation rates are relatively well known. As I mentioned, those gene switching rates are a little bit more difficult to measure, but also can be considered to vary over orders of magnitude depending on what type of gene state switching is occurring. So if we have a 2-gene network model and we simulate this master equation and we look at the steady state co-expression landscape, the bivariate distribution of, in this case, RNA counts that emerges here from this model. Again, I'm plotting this in terms of this 2-dimensional bivariate distribution. Yellow means a lot of cells. And then this is just a representative example of another one of these that's extracted from experimental data. And then we have a question about how do we want to start to integrate our simulated landscapes with experimental landscapes? If I look at the representative example here, it sort of looks okay. But one approach could be to kind of rigorously try to model this. And in fact, we have the probability distribution. So we could look at maximum likelihood estimation and try to really fit parameters and perform model selection. However, we haven't done that there because what we want to do is find a shortcut because if we wanted to start doing this, not just for 2 genes of interest, but more comprehensively across a data set, the expense is a bit overwhelming, although I think there is exciting work to be done in that area. In this work, we're not doing that. We're trying to find a shortcut. So in the rest of what I'll talk about in the remaining time, what we're trying to do is look at distribution shape fingerprints that can reveal information about underlying network interactions. So by training on a set of models, can we figure out what are the best ways to describe these kinds of distributions that we get from simulations, that we get from data, and compare them to each other? And what are the most informative bivariate statistics anyway for these sorts of co-expression landscapes? And so I'll show you, this is ongoing work, but I'll show you what we've done so far. This is work by PhD student Cameron Gallivan. And the first thing that he did was just to build a huge library of model variants where he just computed a bunch of these landscapes. So for example, one library has 81 types of networks. So if you have 4 interactions and 3 types of interaction, repressing, activating, or not interacting, you get 81 networks. And varying kinetic parameters and some plausible ranges. He's looked at on the order of tens to hundreds of thousands of these model variants so far. There are a number of assumptions currently that we're kind of working on trying to improve in our models, but currently some of those are listed here. We're only looking at direct interactions. Currently, we're neglecting indirect interactions in the network. We have only 3 gene expression states. We're considering low or off, basal, and high. We're only looking at intrinsic noise. Currently, we're neglecting fluctuations in protein translation. And as I mentioned, we're computing these in steady state. So we're implicitly making a kind of separation of timescales assumption that these steady state distributions are, and their fluctuations are going to be informative to us of dynamics. Okay, this is just a picture of what, you know, some of his simulated landscapes look like in this library that he's built. The next thing that he did, which was an interesting idea, was to look at how he could organize all the different shapes of these distributions that he was getting out. And he did principal component analysis on the co-expression landscapes. So that's shown here. So what's being shown here is the first 6 principal components. So we're looking at all of the landscapes that he produces in a library. There's confusing coloring on this slide because the genes are colored in blue and red, and down here blue and red simply refers to the sign of the eigenvector of the covariance matrix, and these are, you know, sign invariant eigenvectors, so there's no connection here between those colors and these. But these just sort of emerge from a certain library of models and give us maybe a way to think about what the shapes of those distributions are looking over this large library. We can also generate the same thing from experimental data. And so this is an example of those same types of eigenvectors that we would get if we do this PCA analysis on some experimental data. So this example is from a MoCA data set. This is mouse organogenesis from this paper in 2019. And you get these sort of beautiful different shapes that are emerging. And the first thing that we did when we looked at these was we said, well, our models in some ways sort of capture some of the same features, but they don't look perfect. Maybe we can improve it a little bit. And so we can do another kind of model library. In this library, we've adjusted a couple things. We've increased the sort of range of transcription rates, and we've also accounted for the technical issue of relatively low capture rates in single cell experiments. And we here sort of qualitatively seem to do a little better job of capturing the overall shapes of co-expression that are seen in the measured distributions. Okay, and so the takeaway here is simply that we have sort of this new and interesting way of trying to characterize and quantify the shape space of co-expression landscapes, both from model sets and also from experimental data. And this is published work, and I'm going to just sort of speed through it, but in work that we have done previously, we used these PCA components as a way to look at changes in expression distributions, co-expression distributions over development, and saw sort of signatures of how different kinds of gene pairs with different sorts of interactions appear to show sort of different trajectories. So this is just, again, plotting those different shape components as those eigenvectors. And then if we actually take experimental data and try to project onto those eigenvectors, we see distinctive differences between different types of gene pairs in terms of their evolution of their distributions over development. So that's some stuff that we had published previously. I wanted to sort of briefly show today some of the stuff that we're currently working on. So these are preliminary results, and that's what I'll show now. So what we have now with these libraries is we have a classification problem. We essentially have a whole bunch of different features to describe these distributions. And what we would ultimately like to do is to know if we can classify from those features the interactions, or maybe the whole network logic as envisioned in this cartoon here. And so what I'm showing here is computing the mutual information between the class, whether that be the full network logic or whether that be individual interactions like Y on X, X on Y, et cetera. The mutual information between that class and these continuous features. And the features that we have looked at are plotted on the x-axis. These are some of the ones that we've looked at so far. And so we're just looking at a whole bunch of different, we're sort of fishing here, and we're looking at a whole bunch of different, here these represent what I would call sort of standard bivariate features, summary statistics, if you will, to describe these distributions. We also have a new set of bivariate features, and these are PCA components 1 through 6, and it's difficult to describe what those are, but I'm showing them as a reference on the right-hand side. We've also looked at univariate features as well, and so as a measure of dispersion, we're looking here at the Fano factor of the individual genes X and Y. And so we've found some sort of interesting things in this kind of approach so far. So, for example, in general, for the mutual information between gene X and gene Y, now this is a bit confusing because we use the mutual information between the two genes as a feature, and then we're asking about the mutual information between that feature and the class. And it performs actually relatively poorly. That sort of surprised me, especially compared to something called a Jacquard index, also sometimes referred to as a coexpression index, which is one of the best ones really for this bivariate feature in terms of the information it holds on the full network. And there's some other interesting things here. So one of the things that we noted was that for the self interactions, the dispersion was quite informative. So this is the interactions of Y on itself and X on itself. And that would sort of echo some of the previous work that has been done looking at the fluctuations, size of the fluctuations, and how that relates to the type of self-feedback in a gene. So this is sort of where we're going with this currently, and these are preliminary results. I want to just sort of briefly show what Cameron has done so far. This is where he's trying to look at using this kind of classification based on knowing the ground truth in our simulated library. And so far he has some, I would say, promising results on being able to classify the individual interactions. I say that with a grain of salt because we've thrown a little bit more things at these models in sort of recent weeks, and some of the numbers don't look quite as rosy when we do that. These ones look pretty good. But nevertheless, we think that there's some promise here for being able to classify these interactions. And this is strictly based on a single time point distribution. So it's just saying if you have one distribution, one time point, or one experiment, what's the information content in there on the gene-gene interactions. And then the last result that I'll show before I wrap up here is this one. Again, this is really preliminary, but I wanted to show this because it's sort of where we're trying to go. This is what Cameron has done when he's now applied his method based on these various shape features to trying to classify interactions from experimental data. And so just to briefly walk through what he's looking at here. So this is again from the Mouse Organogenesis Cell Atlas. These are cells in a hematopoiesis lineage. And he's broken them up into a set of something around 450 transcription factors. That's on the left. And on the right, a sort of general top 500 expressing genes set. And he's tried to infer here the target interaction. And these used to be X and Y, and now they say A on B, and I apologize for that. And the other thing I should say is that these really are kind of arbitrary now because we're just throwing these genes at this classification. It's sort of arbitrary which axis they're plotted on, whether it's X and Y or A and B. But what the results are seeming to indicate so far is that in general, while the kind of cross-regulatory interactions between the two genes seem to relatively uniformly sample being either repressive, not present, or activating, we think we're seeing a sort of high amount of self-negative autoregulation, especially in the transcription factor networks, looking just very broadly. That seems to be a network motif that's emerging here from this analysis as one that's more dominant. So this is work in progress, but the takeaway from this is that we think that there is some promise here for using this type of approach, thinking about the distributions, the fluctuations that are revealed by those distributions, and what sort of underlying dynamics we can learn from that. Okay, so I'll wrap up then the second part of the talk. And just to summarize, we tried to think of ways to, in more detail than maybe people had previously done, think about how to describe and quantify co-expression landscape shape. And it seems like we might actually gain something from going beyond even sort of standard bivariate statistical metrics. And so these sort of novel features that we included in our analysis here came from looking at just different sorts of shape features that we got from looking broadly at co-expression landscapes. And as I mentioned, we think that there's potentially underutilized information in these kinds of fluctuations that can be better used maybe if we do it in concert with this type of stochastic modeling. And finally, I think there is still remains huge opportunities here in our sort of omics data deluge. We still have huge challenges remaining for all of us in terms of trying to deal with the difficult computation questions and the best, smartest ways to integrate data with models. So with that, let me thank everybody who has contributed to this work. And I specifically mentioned the 3 lab members of mine, former lab members who did the bulk of the work that I presented today. And I'd like to thank these funding sources. And I would like to thank you for your attention today.